The overall goal of this experiment is to confirm whether pre-synthesized polymeric dyes derived from plant phenols can dye human keratin hairs within an acceptable time frame compared to commercial hair dyes.
This method describes the synthesis of the novel human hair dye. The main advantages of this dyeing technique are that it uses eco-friendly materials, can give rise to various colors, and displays resistant to detergents. Our method shows that polymeric dyes derived from plant phenol polymerization can directly attach to keratin hair surfaces. In addition, the colors and shades are easily variable with metallic salts and pH adjusting agents. The most important step in this method is to polymerize natural phenols with oxidants such as laccases for further dyeing processes. Increased binding ability with polymerization allows natural phenol variants to directly bind to keratin surfaces.
To begin dye preparation, first weigh 0.1 grams of catechol and 0.1 grams of catechin hydrate and add them to 32 milliliters of 100 millimolar sodium acetate buffer. Add 8 milliliters of absolute ethanol and swirl the solution to dissolve the dye. Weigh 10 milligrams of Termetes versicolor laccase and add it to the buffer solution. Mix vigorously and then pour the solution into a square petri dish. Incubate the dish on a shaking incubator at room temperature for 24 hours. Dramatic color change of the solution from clear to dark black will be observed. Pour the solution into a 50 milliliter tube.